In 1946, a young Australian geologist named Reginald Sprigg was doing something utterly mundane, looking for metal deposits in the Ediacara hills of South Australia. He was employed by the government, tasked with surveying remote areas for uranium, copper, anything that might be economically valuable in the post-war boom. It was practical work, unglamorous, the kind of geology that pays bills but doesn't make headlines. He stopped for lunch one day, sitting on an outcrop of reddish sandstone, and noticed something odd. The rock surface beneath him wasn't smooth. It was covered with circular impressions, like fossilized dinner plates pressed into ancient mud. Radial ridges extended from central points. Some were a few centimeters across. Others were larger, nearly a meter in diameter. Sprigg knew fossils. He'd studied paleontology, and he knew these rocks were old, far older than the Cambrian Period, which had begun roughly 540 million years ago. These rocks dated to the Precambrian, a vast span of Earth history that geology textbooks of the time described as essentially lifeless. Bacteria, perhaps, maybe some algae, but nothing complex, nothing you could see with the naked eye, nothing that would leave fossils the size of dinner plates. Yet here they were, dozens of them, hundreds. Once he started looking carefully. Strange, soft-bodied impressions preserved in fine-grained sediments that had once been seafloor mud. Organisms that had lived, died, and been buried before the Cambrian Explosion. Before trilobites, before the sudden appearance of complex life that Charles Darwin himself had called one of the greatest challenges to his theory of evolution. Sprigg collected samples. He brought them to paleontologists at Australian universities. He presented them at conferences. And the response was almost universally dismissive. These couldn't be Precambrian organisms, the experts said. Precambrian rocks didn't contain complex fossils. Therefore, these must be something else. Misidentified jellyfish from younger rocks. Inorganic sedimentary structures that only looked biological. Artifacts of weathering or mineral precipitation. Anything but what Sprigg was claiming. A previously unknown world of life that existed before animals as we understand them. It took decades for the scientific establishment to accept what Sprigg had found. But eventually, similar fossils turned up in other locations. Newfoundland, on the eastern coast of Canada. Namibia, in southwestern Africa. The White Sea region of Russia. Charnwood Forest in England. Sites scattered across multiple continents, all preserving the same types of strange impressions, all from rocks dated to between roughly 635 and 540 million years ago. This wasn't misidentification. This was real. An entire period of Earth history, now called the Ediacaran Period, named after those hills where Sprigg made his discovery, had been filled with complex multicellular organisms. They had thrived for nearly 100 million years. And then, at the boundary between the Ediacaran and the Cambrian, they had almost completely disappeared, leaving only these enigmatic impressions and a scientific mystery that remains unsolved today. If you're intrigued by this story, if you want to explore more mysteries from deep time, please subscribe. Your support makes this work possible. There's much more to discover, and I'm grateful you're here for it. The Ediacaran period lasted from 635 to 540 million years ago. To put that in perspective, the entire age of dinosaurs, from their origin to their extinction, spans roughly 165 million years. The Ediacaran lasted nearly 100 million years, more than half as long as the age of dinosaurs. This wasn't a brief experiment. This was a substantial chapter in the history of complex life. And yet, until Sprigg's discovery, we had no idea it existed. The organisms left no descendants that we can clearly identify. They built no shells or bones that would preserve easily. They vanished so completely that for most of scientific history, we thought the Precambrian was a barren prelude to the sudden explosion of life in the Cambrian. Now we know better. The Ediacaran was filled with life, strange life, life that doesn't fit our categories, life that raises fundamental questions about what it means to be an organism, what strategies evolution tried and discarded, and why the particular forms of life we're familiar with came to dominate while these earlier experiments disappeared. Let me introduce you to the most famous Ediacaran organism, Dickinsonia. Picture an oval disc lying flat on the ancient seafloor. It ranges in size from a few millimeters to over a meter in length, dinner plate to coffee table. The surface shows a quilted pattern with repeating segments running perpendicular to the long axis of the body, giving it a ribbed appearance like a corrugated roof or an air mattress. When paleontologists first examined Dickinsonia fossils, the instinct was to fit them into known categories. The segments suggested it might be related to annelid worms, earthworms, leeches, marine polychaetes, which have segmented bodies. Or perhaps an early arthropod, related to the trilobites and other jointed-legged animals that would dominate Cambrian seas. But the more closely scientists looked, the less sense those interpretations made. In true segmented animals, the segments correspond to internal structures. An earthworm's segments contain repeated sets of organs, muscles, nerves, blood vessels, excretory structures. The segmentation is functional. Each segment is a modular unit with specific anatomical components. Dickinsonia's segments don't appear to work that way. They seem to be just external grooves, surface texture with no obvious internal correlation. There's no evidence of repeated organ systems, no nerve cords running the length of the body, no signs of a gut or a circulatory system. Just segments that divide the body into repeating units without clear functional purpose. Perhaps it was a flatworm. Modern flatworms are bilaterally symmetrical. Left and right sides mirror each other, just like Dickinsonia. They're also flat, as the name suggests, and they crawl along surfaces. But flatworms have heads. They have sensory organs concentrated at one end, eye spots, chemoreceptors, structures that allow them to orient and navigate. Dickinsonia shows no head, no concentration of sensory structures, no obvious front or back beyond the axis of symmetry. Maybe a jellyfish? Some modern jellyfish, when they settle on the seafloor after death, create circular impressions that might superficially resemble Dickinsonia. But jellyfish are radially symmetrical, like a wheel with spokes radiating from a central hub. Dickinsonia is bilaterally symmetrical, and jellyfish have mouths, typically on the underside, surrounded by tentacles. Dickinsonia fossils show no mouth, no tentacles, no features you'd associate with the cnidarian body plan. Could it be a plant? Or rather, could it be a photosynthetic organism, something like a giant algae? The flat form maximizes surface area for light absorption. But algae don't show bilateral symmetry. They don't have the kind of segmented structure Dickinsonia displays. And critically, Dickinsonia fossils have been found in deep water sediments, far below the depth where sunlight penetrates. If it was photosynthetic, what was it doing in the dark? Perhaps a fungus. Fungi can grow in sheet-like forms, spreading across surfaces and absorbing nutrients directly through their tissues. Could Dickinsonia be a giant marine fungus? It's possible, but fungi are typically decomposers, feeding on dead organic matter. The Ediacaran seafloor environment doesn't seem consistent with fungal ecology as we understand it. And fungi don't generally show the kind of regular segmented patterning that Dickinsonia displays. For decades, scientists debated. Every hypothesis ran into problems. Every attempt to slot Dickinsonia into a known group of organisms encountered contradictions. It had features that seemed animal-like, but lacked defining characteristics of animals. It resembled plants in some ways, but wasn't a plant. It had fungal aspects, but didn't fit that kingdom either. Then, in 2018, a team of researchers led by Ilya Bobrovsky analyzed chemical biomarkers preserved in Dickinsonia fossils from Russia. They were looking for lipids, fat molecules, that might reveal something about the organism's biochemistry. Specifically, they were searching for cholesterol. Cholesterol is a lipid molecule that animals produce, but plants and fungi don't. If Dickinsonia contained cholesterol, that would be strong evidence for animal affinity. It would tell us that despite its strange morphology, despite the lack of clear anatomical features, Dickinsonia was biochemically an animal. The results were published in Science. Dickinsonia fossils contained cholesteroids, compounds derived from cholesterol. The ratios and types of these molecules suggested animal metabolism. Not plant, not fungus, not bacteria. Animal. This was a breakthrough. After decades of uncertainty, we finally had chemical evidence placing Dickinsonia in the animal kingdom. But that evidence came with caveats. The specific cholesteroid ratios were unusual, not quite matching modern animal biochemistry. The patterns suggested that while Dickinsonia was probably an animal in the broadest sense, a multicellular heterotroph with differentiated tissues, its metabolism was distinct from anything alive today. So we're left with a paradox. Dickinsonia was probably an animal, but it was an animal unlike any animal we're familiar with. It had no head, no gut, no clear organ systems. It was flat to the point of being two-dimensional. It moved, based on trace fossil evidence we'll discuss later, but moved so slowly that its locomotion was more like oozing than walking or swimming. It lived for tens of millions of years, apparently successfully, in oceans that would be unrecognizable to modern marine organisms. This is where the Ediacaran becomes philosophically interesting. We tend to think of evolution as a process that leads inevitably to the organisms we see today. Fish evolved into amphibians, amphibians into reptiles, reptiles into mammals and birds. A linear progression from simple to complex, from ancient to modern. We imagine that if we go back far enough, we'll find primitive versions of modern animals, early fish, proto-mollusks, ancestral arthropods. But Dickinsonia isn't a primitive version of anything. It's not an early worm or an early flatworm or an early anything we can identify. It's something else entirely. A design that evolution tried extensively. The fossil record shows Dickinsonia and related forms were abundant and widespread, and then abandoned. A successful strategy for nearly 100 million years that left essentially no descendants. This tells us something important about evolution. It's not a directed process with a predetermined endpoint. It's an exploration of possibility space. Most explorations lead nowhere, at least in the long term. They work for a while, Then conditions change, and what worked becomes obsolete. The lineages we see today, the fish and insects and mammals and flowering plants, aren't the inevitable products of evolution. They're the survivors of countless experiments, most of which ended in extinction. Dickinsonia was one of those experiments. It worked beautifully for its time. Then its time ended. Now, let me introduce you to another Ediacaran organism that challenges everything we think we know about life: Charnia. This fossil was discovered not by a professional geologist, but by a teenage boy named Roger Mason, who in 1957 found it in Charnwood Forest in England, an area that geologists had confidently declared too old to contain complex fossils. Mason was 15 years old. He was rock climbing, looking for interesting formations, and he noticed a strange pattern on a rock surface, a long, feather-like structure with a central axis and branches extending alternately on either side. It looked, frankly, like a fern frond, like something you'd see in a forest, not something that should be fossilized in 600-million-year-old marine sediments. He told his teacher. His teacher contacted geologists, and once again the initial response was dismissive. This couldn't be a Precambrian fossil. It must be a younger specimen, somehow mixed into older rocks, or an inorganic structure, a pattern created by mineral precipitation or sedimentary processes that mimicked a biological form. But Mason was right. Charnia was real, and it was Ediacaran. And it was, in some ways, even stranger than Dickinsonia. Charnia looks like a plant. The resemblance to fern fronds is striking. It has a central stalk, often reaching 2 feet or more in length, with branches extending on either side. The whole structure is anchored to the seafloor by a disc-like holdfast, just like many modern marine algae. The branching pattern maximizes surface area, which is exactly what you'd expect from a photosynthetic organism trying to capture as much light as possible. There's just one problem. Charnia fossils have been found in rocks representing deep water environments. Water depths of hundreds of feet, possibly more. Depths where no light penetrates, where photosynthesis is impossible. So if Charnia wasn't photosynthesizing, what was it doing with all that surface area? The answer, as best as scientists can determine, is osmotrophy. That's a technical term for a simple concept: absorbing dissolved nutrients directly from the surrounding water. No mouth, no gut, no process of ingestion and digestion. Just a living surface that pulls organic molecules, carbon compounds, nitrogen, phosphorus, out of solution. This is fundamentally different from how modern animals feed. We eat things. We have mouths, digestive tracts, the whole machinery of breaking down food into absorbable components. Even filter feeders like sponges and clams are actively capturing particles, drawing water through their bodies and straining out suspended food. Osmotrophy is passive. You just sit there, maximize your surface area, and absorb whatever's dissolved in the water around you. For this strategy to work, the water needs to be rich in dissolved organics. Much richer than modern oceans, where dissolved organic matter is relatively dilute. If you tried to survive by osmotrophy in today's ocean, you'd starve. There simply aren't enough nutrients in solution to support a large organism. But the Ediacaran ocean was apparently different. The chemistry was different. The ecology was different. And in that different ocean, osmotrophy was viable. So viable that Charnia grew to substantial sizes and existed in dense communities. sometimes hundreds of individuals in close proximity, all competing for dissolved nutrients flowing past in ocean currents. Now here's where it gets truly strange. Charnia's branching pattern follows fractal geometry. Each branch subdivides into smaller branches, which subdivide into still smaller branches, following a mathematical rule that repeats at different scales. This is the same geometry you see in ferns, in trees, in river drainage networks, in lightning bolts, in your lungs and circulatory system. Fractal branching is an extremely efficient way to pack maximum surface area into minimum volume. It's a solution to the problem of how to create a large interface with the environment without requiring vast amounts of structural material. Nature has discovered this solution repeatedly in different contexts because it's mathematically optimal. But here's what makes Charnia remarkable. It was using fractal geometry 600 million years ago, before plants had colonized land and invented trees, before animals had evolved complex organ systems with fractal circulatory networks. Charnia represents one of the earliest known examples of evolution discovering this geometric principle. And Charnia wasn't unique. Many Ediacaran organisms show similar fractal branching patterns. Scientists call them rangemorphs, named after the mistaken point site in Newfoundland, where they were first studied in detail. These organisms represent an entire design strategy. Fractal osmotrophs, living by absorbing nutrients from seawater, using geometry that wouldn't look out of place in a modern calculus textbook. The implications are staggering. These organisms had no brains, no nervous systems, no capacity for anything we'd recognize as thought or planning. Yet they grew according to mathematical principles that human beings took millennia to understand. Fractal geometry wasn't invented by Benoit Mandelbrot in the 1970s. It was discovered by evolution hundreds of millions of years earlier and implemented in living tissue that extracted nutrients from Precambrian oceans. This tells us something profound about the relationship between mathematics and biology. The patterns we think of as abstract, fractals, symmetries, optimal packing arrangements, are actually deeply embedded in the physical world. Evolution doesn't do math in any conscious sense, but it explores design space, and some regions of that space correspond to mathematical principles. When evolution finds those regions, it produces organisms that embody mathematical elegance without understanding it. Charnia embodied fractal geometry. It maximized surface area using branching algorithms. It did calculus with cells. And then, like Dickinsonia, it vanished. The entire rangemorph design disappeared from the fossil record at the end of the Ediacaran, leaving only these impressions and the questions they raise about the roads not taken in evolution. Let's talk about what the Ediacaran seafloor actually looked like, because understanding the environment is crucial to understanding the organisms. When you picture an ancient ocean, you probably imagine something like a modern ocean, but with different creatures. Blue water, sandy or rocky bottom, maybe some kelp or coral equivalents. The basics are the same, only the details differ. The Ediacaran seafloor was not like that. It was fundamentally alien in ways that would be immediately obvious if you could visit it. First, the substrate. In modern oceans, sediments are loose and mobile. Currents move sand around. Burrowing organisms churn through mud. The seafloor is active, constantly being reworked by physical and biological forces. In the Ediacaran, the seafloor was stable, covered with thick microbial mats, layered communities of bacteria and other microorganisms that bound sediment particles together. These mats were cohesive, almost leathery in texture. They covered vast areas, stabilizing the substrate and creating a fundamentally different kind of seafloor than exists anywhere in modern oceans, except in extreme environments where animals can't survive. These microbial mats were more than just substrate. They were food. Many Ediacaran organisms appear to have grazed on the mats, scraping up bacterial biomass. Others anchored to the mats, using them as attachment points. The mats were the foundation of the entire ecosystem. The primary producers, the habitat, the food source, all in one. Second, the water chemistry. Oxygen levels were lower than today, perhaps 10 to 15% of modern atmospheric levels. This seems like it would be a limitation, but for the organisms that evolved in those conditions, it was normal. They were adapted to extract enough oxygen from low concentrations. Many were thin or highly branched, specifically to maximize the surface area available for oxygen diffusion. The water also contained much higher concentrations of dissolved organic matter than modern oceans. Dead cells, fragments of microbial mats, whatever organic molecules were produced by photosynthetic bacteria in shallow water, all of this material dissolved and circulated through the ocean rather than being quickly consumed by scavengers and decomposers. The Ediacaran Ocean was, in a sense, a dilute organic soup. Third, the absence of certain organisms. No fish, no crustaceans, no mollusks, no echinoderms. None of the groups that dominate modern marine ecosystems. And critically, no predators. As far as we can tell from the fossil record, nothing in the Ediacaran was hunting and eating other large organisms. This is perhaps the most alien aspect of the Ediacaran world. In modern oceans, predation structures everything. Where you live, how you move, what your body looks like. All of it is shaped by the need to avoid being eaten or to successfully hunt. Remove predation, and ecology becomes fundamentally different. Ediacaran organisms didn't need armor. They didn't need to hide or flee. They didn't need to move quickly or sense approaching threats. They could be soft-bodied, slow-moving, and conspicuous because nothing was hunting them. This allowed body plans that would be suicidal in a world with predators. Imagine lying flat on the seafloor like Dickinsonia, exposed, vulnerable, moving at millimeters per hour. In a modern ocean, you'd be eaten immediately. In the Ediacaran Ocean, you were fine. The rules were different. This peaceable kingdom lasted for nearly 100 million years. And then, at the end of the Ediacaran, something changed. Predation appeared. And when it did, the entire ecosystem was revolutionized. Most of the Ediacaran body plans went extinct, and the Cambrian Explosion began. But we're getting ahead of ourselves. Before we discuss the end, we need to understand more about what existed. Because the Ediacaran biota was far stranger than just Dickinsonia and Charnia. There were dozens of distinct forms, each raising its own questions, each representing an evolutionary experiment that has no clear parallel in the modern world. Consider the organism called Spriggina, named after Reginald Sprigg. It's roughly elongate, a few centimeters long, with what appears to be a curved or crescent-shaped head shield at one end and a series of repeated segments along the body. When paleontologists first described it, the immediate interpretation was that it must be related to trilobites, those iconic Cambrian arthropods with segmented bodies and hard exoskeletons. But look more closely at Spriggina and problems emerge. The segments on the left side of the body don't align with the segments on the right side. They're offset, creating a zigzag pattern down the midline. This is called glide reflection symmetry, and it doesn't occur in any known segmented animal. In true segmented animals, annelids, arthropods, vertebrates, the segments are paired. Left and right sides match. The segmentation reflects underlying anatomical organization, with organs and structures repeated in each segment. In Spriggina, the offset segments suggest something different. Maybe it wasn't truly segmented at all. Maybe what looks like segmentation is just surface texture, grooves that don't correspond to internal divisions. Or maybe Spriggina represents a type of segmentation that evolution tried and discarded. A body plan that worked for a while, but was ultimately outcompeted by the more familiar types of segmentation that persist in modern animals. We don't know. Spriggina is another enigma, another fossil that doesn't quite fit our categories. And it's not alone. The Ediacaran is full of forms like this. Organisms that look almost familiar, that seem to be on the verge of fitting into known groups, but that ultimately resist classification. There's Kimberella, which we'll discuss in more detail shortly. That might be related to mollusks, but might be something else entirely. There's Tribrachidium, with its bizarre threefold symmetry that makes no sense by the rules of modern biology. There's Pteridinium, which looks like it's been folded into a complex three-dimensional origami structure for reasons no one understands. Each of these organisms was successful in its time. Each left fossils in multiple locations across different continents. Each persisted for millions of years. These weren't failures or evolutionary dead ends in any meaningful sense. They were viable life strategies that worked in the Ediacaran world. But they didn't survive the transition to the Cambrian. And that fact, that an entire assemblage of successful organisms could disappear when conditions changed, tells us something important about the contingency of evolution. Success is always relative to circumstances. The Ediacaran organisms were superbly adapted to late Precambrian oceans. Low oxygen, mat-covered substrates, no predation, high dissolved organics. Those adaptations were perfect for that world. But when the world changed, those adaptations became irrelevant or even detrimental. And organisms that had thrived for nearly 100 million years went extinct. This is a lesson worth absorbing. Adaptation doesn't prepare you for change. It optimizes you for current conditions. If conditions remain stable, that's fine. But conditions rarely remain stable over geological timescales. And when they change, the most perfectly adapted organisms might be the most vulnerable, because they've specialized for a world that no longer exists. The Ediacaran organisms specialized for a world that was temporary. They couldn't know that. Evolution doesn't plan for the future. It only responds to the present. And in the present they inhabited, they were successful. Then the present changed, and success ended. Let me tell you about an organism that might be the most important fossil in the Ediacaran, Kimberella. It's not the largest, typically just a few centimeters long. It's not the most abundant. It's not the strangest looking. But it might be the one that tells us the most about the origins of complex animal life. Kimberella fossils show an oval body with a distinct front and back. Unlike Dickinsonia's ambiguous symmetry, Kimberella clearly has directionality. One end is different from the other. There's a concentration of features at what appears to be the anterior, possibly a mouth region, possibly sensory structures. though preservation doesn't allow certainty. But here's what makes Kimberella extraordinary. It's associated with trace fossils, scratch marks on bedding surfaces, grooves and furrows that appear to have been made by the organism as it moved. And these aren't random marks. They show patterns consistent with feeding behavior. The traces suggest Kimberella was grazing on microbial mats. It would crawl slowly across the seafloor, scraping up bacterial biomass with some kind of feeding structure. Perhaps a muscular foot like modern mollusks use. Perhaps something entirely different. As it moved, it left behind these characteristic traces, parallel grooves where it had scraped the mat surface, areas where the mat was disturbed or removed. This is behavioral evidence. We're not just seeing what Kimberella looked like, we're seeing what it did. And what it did looks remarkably like what some modern animals do. Specifically, it looks like what primitive mollusks do, grazing on biofilms using a radula, that ribbon of tiny teeth that snails and their relatives use to scrape food from surfaces. Could Kimberella be related to mollusks? Could it be a stem group mollusk, representing the lineage before true mollusks evolved, but after it had split from other animal groups? The evidence is tantalizing, but not conclusive. The body morphology is consistent with mollusk affinity. The feeding traces are consistent with mollusk-like behavior. But we don't have shells. We don't have radula preserved. We don't have the definitive anatomical features that would prove the connection. What we can say is this: Kimberella was a bilaterian. It had bilateral symmetry, left and right sides mirroring each other. It had directed movement, a front and a back, associated with forward locomotion. It had feeding behavior that required some level of sensory motor coordination. It could detect where food was and move toward it. These are characteristics we associate with animals, specifically with the vast group of animals called bilaterians, that includes everything from insects to fish to humans. You are a bilaterian. Kimberella was probably a bilaterian. That means somewhere in deep time, your lineage and Kimberella's lineage shared a common ancestor. The connection is distant, over 500 million years distant, but it's real. The bilateral body plan, the directed movement, the ability to sense and respond to the environment, these features arose before Kimberella, and they've been passed down through countless generations to reach you. This makes Kimberella more than just another weird Ediacaran organism. It's potentially a representative of your deep ancestry. Not a direct ancestor. The line from Kimberella to you isn't straight, and it's possible Kimberella's lineage ended without descendants. But at minimum, Kimberella represents the kind of organism your ancestors were in the late Ediacaran. Bilaterians with directed movement, living on ancient seafloors, grazing on microbial mats, beginning the evolutionary trajectory that would lead to vertebrates and eventually to you. The trace fossils Kimberella left are also important for another reason. They show the beginning of bioturbation. That's the technical term for organisms disturbing sediments. When Kimberella scraped the seafloor, it broke up microbial mats. It churned the surface layer of sediment. It mixed oxygenated water into sediments that had been anoxic. This seems trivial. So, what if some small organism scratched the seafloor? But bioturbation, once it began, had cascading effects. Microbial mats that had stabilized sediments for hundreds of millions of years were now being disrupted. Nutrients that had been locked in those mats were being released. Sediment chemistry was changing as oxygen penetrated deeper. And as more organisms evolved the ability to burrow and disturb sediments, bioturbation intensified. By the early Cambrian, seafloors that had been stable and mat-covered for eons were being completely reworked by biological activity. The entire physical and chemical structure of marine sediments changed. For organisms adapted to mat-covered seafloors, organisms like Dickinsonia and the rangemorphs, this was catastrophic. Their habitat was being destroyed. Attachment points disappeared. The stable substrates they relied on became loose and mobile. It's an example of ecosystem engineering, organisms modifying their environment in ways that affect other species, often negatively. Humans are ecosystem engineers too. We modify landscapes, alter atmospheric chemistry, change the availability of resources. Most species don't benefit from our modifications. Many go extinct as a result. We're recapitulating, in some sense, the dynamics of the Ediacaran Cambrian transition. One group of organisms changing the world, and other groups unable to cope with the changes. Kimberella didn't intend to destroy Ediacaran ecosystems. It was just feeding, just living its life. But the side effects of its behavior, multiplied across millions of organisms over millions of years, transformed the world. That's how ecological change usually happens. Not through dramatic catastrophes, but through incremental modifications that compound over time until the world has fundamentally shifted. Now let's talk about an organism that embodies one of the strangest aspects of Ediacaran biology, Tribrachidium. This is a small, disc-shaped fossil, typically a few centimeters across. It sat on the seafloor, probably sessile, permanently attached to one spot. At first glance, it might not seem remarkable. Then you notice the symmetry. Tribrachidium has threefold radial symmetry. 3 curved arms radiate from a central point, dividing the body into 3 equal sections. Each arm appears identical, creating a pattern that repeats every 120 degrees. Threefold symmetry is extraordinarily rare in biology. Go outside and look at organisms. Look at animals, plants, fungi, anything you can find. Count the symmetries. You'll see bilateral symmetry, left and right mirror images. You'll see radial symmetry with multiples of 4, 5, or 6. Flowers often have 5 petals. Starfish have 5 arms. Many jellyfish have 4-fold or 8-fold symmetry. Even radiolarians, microscopic planktonic organisms, tend toward multiples of 3, 6-fold, 9-fold, 12-fold. But pure 3-fold symmetry? Almost non-existent. There are mathematical and developmental reasons for this. Bilateral symmetry emerges naturally from directed movement. You need a front and back, which creates left and right as mirror images. Radial symmetry emerges from being sessile or drifting, where no direction is privileged. And if you have radial symmetry, higher multiples, 4, 5, 6, provide better spatial coverage than 3. Yet Tribrachidium had 3-fold symmetry. Hundreds of fossils have been found, from multiple sites, all showing the same pattern. This wasn't a developmental abnormality. This was a stable body plan that persisted for millions of years. What was it? Some scientists have suggested it might be related to echinoderms, the group that includes starfish, sea urchins, sea cucumbers. Echinoderms have 5-fold symmetry as adults, but their larvae are bilaterally symmetrical. Perhaps Tribrachidium represents an early experiment with radial symmetry before the 5-fold pattern became fixed. But there's no direct evidence for this. Tribrachidium shows no features that clearly link it to echinoderms. The threefold symmetry doesn't seem to be a step toward fivefold symmetry. It's just threefold, consistently and repeatedly, as if this was a viable design in its own right. Other scientists think Tribrachidium isn't related to any modern group. It's another Ediacaran experiment that went nowhere. Evolution tried threefold symmetry, found it worked well enough to persist for millions of years, and then abandoned it when other designs proved more successful in the changed conditions of the Cambrian. This is humbling. We think of biological form as being constrained by mathematical and physical laws. Certain patterns emerge because they're optimal. Bilateral symmetry for movement, radial symmetry for sessile filter feeding, fivefold symmetry for echinoderms. Because, well, we're not entirely sure why echinoderms settled on 5, but they did, and it must have worked. Tribrachidium suggests that the space of viable body plans is larger than we think. Threefold symmetry seems suboptimal by modern standards. It doesn't provide the spatial coverage of higher multiples, and it doesn't have the efficiency of bilateral symmetry. Yet, it worked. Tribrachidium existed, apparently successfully, for millions of years. Maybe threefold symmetry was optimal for conditions that no longer exist. Maybe it provided some advantage in the Ediacaran environment that became irrelevant in the Cambrian. Or maybe it was just good enough, not optimal, but viable. And viability is all evolution requires. You don't have to be the best possible design. You just have to be good enough to survive and reproduce. Tribrachidium was good enough. Then the world changed, and good enough wasn't enough anymore. The threefold body plan disappeared, and with it, whatever advantages or peculiarities it represented vanished from the history of life. Let me take you to Mistaken Point in Newfoundland, Canada, because what scientists have found there is perhaps the most alien ecosystem ever documented on Earth. The site preserves a deep-water Ediacaran community, probably several hundred feet below the ocean's surface. No light reaches this depth. Photosynthesis is impossible. In modern oceans, deep water is sparsely populated, a few scattered organisms eking out existence in a food-poor environment. Mistaken Point was the opposite. The bedding plains, flat rock surfaces representing ancient seafloors, are covered with fossils, thousands of them per square meter. Dense communities of rangeomorphs, those fractal branching organisms we discussed earlier, standing upright like a forest of mathematical ferns. A forest in the deep ocean, in the dark, with no photosynthesis, with no obvious food source. This shouldn't be possible. Modern deep-sea ecosystems are food limited. Organisms survive on marine snow, the constant rain of dead organic matter from surface waters, and on rare bonanzas like whale falls, where a dead whale sinking to the bottom provides a temporary feast. Productivity is low. Populations are sparse. Large organisms are rare. Yet the Mistaken Point assemblages show large organisms, some over 2 feet tall, in extraordinary densities. Something was supporting this biomass. The water flowing through this deep-sea forest must have been rich in dissolved organic matter, much richer than modern deep oceans. Where was that dissolved organic matter coming from? The most plausible source is productivity in shallow water, photosynthetic bacteria, and early algae producing organic compounds, some of which dissolved and were transported to deeper water by currents. But even accounting for that, the concentrations must have been high, much higher than anything we see today. Why? What was different about Ediacaran ocean chemistry that allowed dissolved organics to accumulate to such high levels. One possibility is lower oxygen. In modern, well-oxygenated oceans, bacteria quickly decompose organic matter, breaking it down into inorganic nutrients. The residence time of dissolved organics is short. They're produced and consumed rapidly. In low-oxygen environments, decomposition is slower. Organic matter persists longer in dissolved form, accumulating to higher concentrations. The Ediacaran Ocean had lower oxygen levels than today. This would slow bacterial decomposition and allow dissolved organics to build up. The result? An ocean that was, compared to modern oceans, an organic soup. Dilute, but rich enough to support organisms that made a living by absorbing nutrients from solution. Another possibility involves the absence of efficient consumers. Modern oceans have countless filter feeders: sponges, bivalves, tunicates, zooplankton that strain particles and dissolved organics from water. These organisms are so efficient that they prevent dissolved organics from accumulating. In the Ediacaran, such efficient consumers might not have existed or were much less abundant. Nutrients could build up because nothing was removing them quickly. Either way, the mistaken point assemblages tell us that the Ediacaran deep ocean was fundamentally different from modern deep oceans. It was productive enough to support dense forests of large organisms using a feeding strategy, osmotrophy, that doesn't work in modern marine environments. This was an ocean with different rules, different chemistry, different ecology. And those rangemorph forests reveal something else: competition. The fossils aren't randomly distributed, they're structured. Smaller organisms cluster in areas of high water flow, where nutrient delivery would be maximized. Larger organisms are more evenly spaced, suggesting they interfere with each other, perhaps by depleting nutrients in the surrounding water, creating a zone where other organisms can't get enough food. Some species occur only in specific depth ranges or sediment types. There's ecological zonation, different species exploiting different niches, just like in modern ecosystems. This wasn't a random collection of weird organisms. This was an organized community with competitive interactions, resource partitioning, and spatial structure. The rangemorphs might look alien, but they were doing what organisms do: competing for resources, adapting to local conditions, creating ecological patterns. The underlying principles were the same as today. The specific forms were different, but the ecology was recognizable. This matters because it tells us that by the late Ediacaran, roughly 570 to 560 million years ago, complex ecology had evolved. Not just individual organisms, but communities of interacting organisms, structured by competition and environmental gradients, functioning as ecosystems in ways we can analyze and understand. And then, in just a few million years, those ecosystems vanished. The rangemorph forests disappeared. The deep water assemblages ended. By the early Cambrian, organisms using the rangemorph strategy, fractal osmotrophs standing upright in water flow, were gone from the fossil record. Why? What happened? Several factors likely contributed. Ocean chemistry changed. Oxygen levels rose, possibly reducing dissolved organic concentrations. The evolution of more efficient consumers might have depleted nutrients that rangemorphs depended on. And critically, bioturbation destroyed the stable substrates that rangemorphs needed for attachment. Rangemorphs were anchored to the seafloor by holdfasts. disc-like structures that attached to microbial mats. When burrowing organisms began breaking up those mats, rangemorphs lost their attachment points. A forest can't exist without ground to root in. When the ground disappears, the forest dies. This is ecosystem collapse driven by habitat modification. The organisms causing the modification, burrowers churning through sediments, weren't intentionally destroying rangemorph habitat. They were just living. But the side effects of their activity changed the physical structure of the seafloor, and organisms adapted to the old structure couldn't survive the change. Modern humans are causing similar changes. We're modifying habitats, clearing forests, warming oceans, acidifying seawater, in ways that don't directly target other species, but destroy the conditions those species require. The result is extinction, not because we're hunting species to extinction, but because we're changing the world faster than they can adapt. The Ediacaran Cambrian transition is a warning from deep time. Change the fundamental conditions of an ecosystem, and the organisms adapted to old conditions will disappear, no matter how successful they've been. Duration of success doesn't confer immunity to change. The rangemorphs thrived for tens of millions of years. Then the world shifted and they vanished. Let's talk about something that will make you reconsider what we mean by animal. In the late Ediacaran, possibly overlapping with the earliest Cambrian, organisms called Claudina and Namacalathus appeared. These are small, typically millimeters to centimeters long, and they did something no previous organism had done. They built mineralized skeletons. Claudina constructed a tube from calcium carbonate, growing it in segments that nested inside each other like stacked cones. Namacalathus built a cup-shaped structure, also mineralized, with a distinctive goblet form. These are the first biomineralizers, organisms that precipitated minerals from seawater and incorporated them into structural tissues. This might not sound revolutionary. Shells are common. Most marine animals have some form of mineralized structure. But before Claudina and Namacalathus, nothing did. For hundreds of millions of years, life had been entirely soft-bodied. Now, suddenly, organisms were building hard parts. Why? What changed? The most compelling explanation involves predation. And here's where the story takes a dark turn. Some Claudina fossils show circular holes drilled through the tube walls. The holes are too regular to be accidental damage. They appear to have been made by another organism, a predator that bored through Claudina's protective skeleton to access the soft tissue inside. This is the first definitive evidence of predation in the fossil record. Before this, there are no unambiguous signs that organisms were hunting and eating each other. The Ediacaran, for most of its duration, appears to have been a world without predators. Organisms absorbed nutrients from water or sediment, or photosynthesized, or grazed on microbial mats. But they didn't hunt. That changed in the terminal Ediacaran. Some organism, we don't know what, evolved the ability to attack other organisms, drill through their defenses, and consume them. This is a profound ecological shift. It means the evolution of predatory behavior, with all its implications. Once predation exists, everything changes. Prey organisms are under intense selection pressure to develop defenses. Shells, spines, toxins, burrowing behavior, speed, anything that reduces the probability of being eaten. And predators are under pressure to overcome those defenses. Stronger drilling structures, chemical secretions that dissolve shells, better sensory systems to locate prey. faster movement to pursue. This creates an evolutionary arms race. Each innovation in defense drives counter-innovations in offense and vice versa. The result is rapid diversification as lineages split into predator and prey specializations, each elaborating on strategies that work. The Cambrian Explosion, that sudden proliferation of animal diversity that begins roughly 540 million years ago, is often explained as the result of this arms race. Once predation appeared, it triggered cascading evolutionary changes that produced the diversity of body plans we see in Cambrian fossils. Eyes evolved to see predators or prey. Armor evolved to resist attack. Speed evolved to escape or chase. Complexity increased because survival demanded it. And in this new, dangerous world, the Ediacaran organisms, slow-moving or sessile, soft-bodied, with no defensive structures, were hopelessly vulnerable. They were adapted to a peaceable world that no longer existed. Some might have persisted in refugia, places where predation pressure was lower. But eventually, they disappeared. The fossil record shows this transition. In the oldest Ediacaran rocks, predation is absent. In the youngest Ediacaran and earliest Cambrian rocks, predation is evident. And the organisms change accordingly. Soft-bodied Ediacaran forms decline. Hard-shelled Cambrian forms proliferate. The world transforms from a garden to a battleground. This transformation happened in just a few million years. Geologically, that's rapid. Biologically, it's an eye blink. And it changed the rules of life on Earth permanently. Before the late Ediacaran, being soft-bodied was fine. After the Cambrian began, being soft-bodied was often fatal. Evolution had crossed a threshold, and there was no going back. Predation, once invented, couldn't be uninvented. The arms race, once started, couldn't be stopped. And organisms that had thrived for nearly 100 million years in the absence of predation found themselves obsolete in just a few million years after predation appeared. This is a lesson about evolutionary contingency. The Ediacaran organisms weren't poorly designed. They were exquisitely adapted to their world. But their world was contingent on the absence of predation. Once predation evolved, everything that depended on that absence became vulnerable. You live in a world shaped by predation. Your eyes exist because predation created selection pressure for vision. Your ability to run, to react quickly, to feel fear, all of these traits were honed by millions of years of predators chasing prey and prey fleeing predators. The world you inhabit is fundamentally different from the Ediacaran world because of one innovation: organisms eating other organisms. That innovation appeared roughly 545 million years ago, and it ended the Ediacaran experiment, replacing it with the Cambrian arms race that continues today. Now let me tell you about an organism that might be the strangest of all. Pteridinium. Imagine a structure that looks like it's been folded from a flat sheet into a three-dimensional form, creating a quilted modular body with multiple axes of symmetry. It's not bilaterally symmetrical. It's not radially symmetrical. It has a symmetry called triradial, three-part, but not like Tribrachidium's threefold radial pattern. This is different, more complex, involving folded surfaces and interlocking modules. Pteridinium reached sizes of over a foot long. It stood upright on the seafloor, anchored by a holdfast. And it's completely unlike anything alive today. There's no modern organism we can point to and say Pteridinium was probably related to that. It's an orphan, an evolutionary experiment that left no descendants we can identify. What was it doing? How did it feed? How did it grow? We don't know. The morphology is so strange that interpreting function is nearly impossible. It might have been an osmotroph, like the rangermorphs, absorbing nutrients from water flowing past. It might have hosted photosynthetic symbionts, though it's been found in deep water deposits. It might have done something we can't imagine, because no modern organism does it. Pteridinium represents the furthest extreme of Ediacaran strangeness. Not just different from modern organisms, but different in ways we struggle to conceptualize. It's like finding a machine from an alien civilization. You can see that it's structured, that it's not random, but you can't figure out what it does because it operates on principles you don't recognize. And it worked. Pteridinium persisted for millions of years. It's found at multiple sites across multiple continents. Whatever it was doing it successfully. Then the Ediacaran ended and Pteridinium vanished, leaving only these enigmatic fossils and the profound reminder that the space of possible life is far larger than the subset of life that currently exists. Evolution has explored vast regions of morphospace, the abstract space of possible body plans that are now empty. Pteridinium occupied one of those regions. So did Dickinsonia and Charnia and Tribrachidium, and the hundreds of other Ediacaran forms we've discovered. They weren't failures. They were viable solutions to the problem of being multicellular. But they were solutions that worked only under Ediacaran conditions. When conditions changed, those solutions became obsolete. The regions of morphospace they occupied were vacated, and evolution, moving forward, explored different regions, the ones that led to trilobites and mollusks and vertebrates, the ones that include organisms alive today. But the empty regions remain, at least as abstract possibilities. Their roads evolution tried and then abandoned. And studying them, studying the Ediacaran organisms, gives us a glimpse of how different life could have been, and perhaps how different it could yet become if conditions changed in ways that favored body plans we haven't seen for 500 million years. Now we need to talk about ice. Because to understand the Ediacaran, you need to understand what came immediately before it, the most extreme climate event in Earth's history. Around 635 million years ago, just before the Ediacaran period began, Earth experienced something so severe, it's called Snowball Earth. Not an ice age, not glaciation confined to the poles. A complete planetary freeze. Ice sheets extending from pole to pole, across every continent, covering every ocean. The entire surface of Earth locked in ice potentially a kilometer thick. Surface temperatures plunging to -50 Celsius or colder for somewhere between 5 and 15 million years. The evidence is unambiguous. Geologists find glacial deposits, distinctive rocks dropped by melting icebergs called dropstones, in sequences that indicate they were deposited at tropical latitudes. They find rocks with scratch marks from glaciers grinding across bedrock, again at latitudes that are now tropical. They find chemical signatures in rocks suggesting that ocean circulation nearly stopped, consistent with surface waters being frozen solid. This event is called the Marinoan glaciation, and it was the last and most severe of several Snowball Earth episodes. The planet had frozen before, but never this completely, never this long. And when it finally ended, it ended catastrophically. Here's what happened. Volcanic activity continued throughout the freeze. Volcanoes don't care if the surface is frozen. They're driven by heat from Earth's interior, and they kept erupting, pumping carbon dioxide into the atmosphere. But with the surface frozen, that CO2 couldn't be removed by the normal weathering processes. The chemical reactions between carbon dioxide, water, and rocks that lock carbon into minerals. So for millions of years, carbon dioxide accumulated. The atmospheric concentration rose higher and higher, 10 times pre-industrial levels, 100 times, maybe more. And eventually, the greenhouse effect became strong enough to overcome the ice albedo feedback, the tendency of ice to reflect sunlight and maintain cold temperatures. The ice began to melt, and once melting started, it accelerated. Less ice meant darker surfaces, land and ocean absorbing more sunlight instead of reflecting it. More absorption meant more warming. More warming meant more melting. It's a positive feedback loop, and it ran away. Within perhaps a few thousand years, geologically instantaneous, Earth went from completely frozen to extremely hot. Surface temperatures might have soared above 50 Celsius. Oceans that had been ice-covered for millions of years suddenly warmed dramatically. The rocks record this transition. Glacial deposits are directly overlain by thick layers of limestone, called cap carbonates, that precipitated rapidly from supersaturated oceans as chemical weathering went into overdrive in the extreme heat. This is one of the most dramatic transitions in the geological record. Freeze to furnace in a few millennia, and it marks the beginning of the Ediacaran period. This matters because complex multicellular life, the Ediacaran biota, appeared after this transition. The oldest definitive Ediacaran fossils date to roughly 570 million years ago, about 65 million years after the Marinoan glaciation ended. That's enough time for evolution to work with, but it's not an enormous amount of time by geological standards. Where did the Ediacaran organisms come from? Did they evolve from scratch after Snowball Earth? Or did their ancestors survive the freeze in refugia? Volcanic hot springs, deep-sea hydrothermal vents, pockets of liquid water beneath ice sheets, and then diversify rapidly when conditions improved? We don't know. The fossil record from before the Marinoan glaciation is sparse and ambiguous. There are hints of complex multicellular organisms in rocks from before Snowball Earth, but nothing definitive. The glaciation itself destroyed much of the pre-existing rock record, grinding it away with continent-spanning ice sheets. We're left with fragments, not a clear picture. But some scientists think Snowball Earth might have accelerated the evolution of complexity. The hypothesis is that extreme environmental stress creates intense selection pressure. Most lineages go extinct, but those that survive do so because they have innovations, genetic or developmental flexibility that allow adaptation to marginal conditions. Maybe the genetic toolkits that later produced Ediacaran body plans evolved initially as survival mechanisms during Snowball Earth. Maybe the ability to form differentiated tissues, to coordinate cell types, to build large multicellular structures. All these capabilities emerged because they helped organisms survive in the tiny habitable zones that persisted during the freeze. And then, when the ice melted and conditions improved dramatically, those same capabilities were repurposed. Evolution took the mechanisms that had been used for survival in extreme conditions and used them to build the diverse forms we see in Ediacaran fossils. The crisis created the tools. The recovery created the opportunity. and the result was an explosion of morphological experimentation. This is speculative, but it's testable in principle. If we could find fossils from immediately before Snowball Earth and compare them to post-glaciation forms, we could see whether complexity increased after the freeze. Unfortunately, such fossils are rare for the reasons I mentioned. But the hypothesis is compelling because it explains a puzzle. Why did complex life appear when it did, and not earlier or later? Maybe because Snowball Earth created conditions, both the stress during the freeze and the opportunities after the thaw. That made complexity advantageous in ways it hadn't been before. And if that's true, then you owe your existence not just to evolutionary processes operating under normal conditions, but to a planetary catastrophe that nearly ended life entirely. Your ancestors, whatever they were, survived the freeze. They adapted to the thaw. They navigated the most extreme climate event in Earth's history and came out the other side capable of giving rise to the Ediacaran biota, which eventually gave rise to Cambrian animals, which eventually gave rise to you. That's a humbling genealogy. You exist because something survived the unsurvivable. Now let's talk about what didn't happen in the Ediacaran, because absences can be as revealing as presences. There's no clear evidence of eyes, no fossilized eye structures, no organisms with obvious visual organs. And this matters because eyes are transformative. In modern ecosystems, vision structures everything. Predators use eyes to hunt. Prey use eyes to flee. Camouflage, warning coloration, bioluminescence. All these phenomena exist because organisms have eyes. Remove vision from an ecosystem, and the ecology becomes fundamentally different. The Ediacaran appears to have been a world without eyes. Organisms might have had light-sensitive cells. Many single-celled organisms have simple photoreceptors that detect light and shadow. But complex image-forming eyes, the kind that allow you to see shapes and movement and other organisms, don't appear until the Cambrian. This has profound implications. If Ediacaran organisms couldn't see each other, they couldn't use visual signals. Coloration would be irrelevant for communication or warning. Camouflage would be unnecessary. Predators, if they existed, would have to find prey using other senses. Chemical detection, mechanoreception, detecting vibrations or water currents. This might explain why the Ediacaran world seems so strange to us. We're visual organisms. We navigate the world primarily through sight. We interpret other organisms based on what they look like. But in a world without vision, those visual cues would be meaningless. Body forms would be shaped by other constraints: hydrodynamics, nutrient absorption, structural integrity, not by the need to look threatening or camouflaged or attractive. Eyes evolved rapidly in the early Cambrian. Multiple lineages independently evolved complex visual systems within a few million years. Why then? What changed? The most likely answer is predation. Once organisms started hunting each other, there was intense selection pressure for detecting prey or predators. Vision provides information at a distance. It allows you to see threats or opportunities before they reach you. In a world with predation, vision is enormously advantageous. So, eyes evolved. And once eyes existed, ecology changed forever. Predators could hunt more efficiently. Prey could flee more effectively. And this drove further innovation. Better vision in predators, better evasion strategies in prey, an arms race that continues to this day. Your eyes, the ones you're using to read or listen to this, are products of that Cambrian arms race. They descended from the first image-forming eyes that evolved roughly 530 million years ago. And those eyes didn't exist in the Ediacaran. The organisms we've been discussing lived in a world without sight, and it made them fundamentally different from organisms in the world that followed. Let me tell you about the seafloor itself, because understanding what happened to it explains much about why Ediacaran organisms disappeared. For hundreds of millions of years, seafloors had been covered with microbial mats, thick, cohesive layers of bacteria binding sediment particles together. These mats stabilized the substrate. They created a firm surface that organisms could attach to, crawl on, or anchor into. Then burrowing evolved. Initially, just shallow burrowing, organisms making simple horizontal traces just beneath the sediment surface. But over time, burrows became deeper and more complex. And as organisms churned through sediments, they broke up the mats. This process, called bioturbation, fundamentally transformed seafloor environments. Burrows allowed oxygen to penetrate deeper into sediments, changing the chemistry. The physical structure of the seafloor changed from stable and mat-covered to loose and mobile. Nutrients that had been locked in mats were released. Sediment grain size distributions changed as fine particles were mixed with coarse particles. By the early Cambrian, most shallow water sediments were heavily bioturbated. Microbial mats still existed in some places, extreme environments where burrowers couldn't survive. But they were no longer the dominant feature of seafloors. The world had changed from mat-covered to bioturbated. For organisms adapted to mat-covered seafloors, this was catastrophic. Rangeomorphs that anchored to mats lost their attachment points. Dickinsonia that crawled on stable mat surfaces found those surfaces disappearing beneath them. Trace fossils from the terminal Ediacaran show organisms struggling, trails that wander seemingly randomly, organisms that appear to have been unable to maintain position as sediments destabilized. It's ecosystem engineering on a planetary scale. Burrowers didn't intend to destroy Ediacaran habitats. They were just feeding, just living in the sediments. But the side effects of their activity changed the physical structure of the seafloor, and organisms adapted to the old structure couldn't survive the change. This is how most extinctions happen. Not sudden catastrophes, but gradual environmental change driven by biological or geological processes. One group of organisms modifies the environment. Other groups, adapted to the old environment, decline. Eventually, they disappear. The Ediacaran organisms weren't killed by a meteor or a volcanic eruption. They were outcompeted and habitat destroyed by organisms better adapted to the changing world. Slow extinction, playing the strange forms of the Ediacaran, had been replaced entirely by the familiar forms of the Cambrian. Now, I need to tell you about something that will challenge your understanding of what success means in evolution. There's a modern organism called Trichoplax adhærens, a placozoan, one of the simplest animals alive. It's just a few thousand cells organized into a flat plate about 2 millimeters across. No mouth, no gut, no nervous system, no muscles, no organs at all beyond 3 cell types. Trichoplax crawls along surfaces and absorbs nutrients through its ventral surface. It reproduces by fission, literally splitting in half. It's so simple that when it was first discovered, scientists thought it might be the larval stage of something more complex. But no, this is the adult form. This is as complex as it gets. For decades, Trichoplax was considered a living fossil, a primitive organism representing what early animals looked like. A window into the deep past, preserved largely unchanged for hundreds of millions of years. Then molecular biology arrived. Scientists sequenced Trichoplax's genome, and what they found was shocking. The genome contains genes for building nervous systems, genes for building muscles, genes for constructing complex body plans. But those genes aren't expressed. They're there in the DNA, functional but turned off. Trichoplax isn't primitively simple, it's secondarily simplified. Its ancestors were more complex, had nervous systems, had muscles, had elaborate anatomy. And then evolution stripped all that away, leaving only the bare minimum necessary for survival. Why would evolution do this? Because complexity is expensive. Brains require enormous amounts of energy. Muscles require energy. Complex developmental programs require regulatory machinery that's costly to maintain. If you're living in an environment where simplicity works, where you don't need to move fast, don't need to sense predators, don't need to compete aggressively, then shedding complexity can be advantageous. You save energy, you reproduce faster. You can survive in low-nutrient environments where complex organisms would starve. Simplicity becomes a competitive advantage. And over millions of years, natural selection favors the simple forms, eventually producing organisms like Trichoplax that look primitive but are actually highly derived. This matters for understanding Ediacaran organisms. When the Ediacaran ended, most of the distinctive body plans disappeared from the fossil record. We interpret this as extinction. But maybe some lineages didn't go extinct. Maybe they simplified. Maybe descendants of Dickinsonia or rangeomorphs still exist, but they're small and cryptic and so simplified that we don't recognize the connection. Maybe they're living in the spaces between sediment grains or attached to the undersides of rocks, too small and simple to leave fossils, unknown to science because they don't attract attention. This is speculative. We have no direct evidence that Ediacaran lineages simplified and survived. But the existence of organisms like Trichoplax tells us it's possible. Lineages can disappear from the fossil record without going extinct. They can evolve to become unfossilizable. They can shed the features that made them recognizable. The absence of Ediacaran forms in post-Cambrian rocks doesn't necessarily mean total extinction. It might mean transformation into something so different that we've lost track of them. And that's a humbling thought, that the strange organisms we've been discussing might still be with us, hidden in plain sight, having survived by becoming small and simple and invisible. Trichoplax has been around for hundreds of millions of years. It survived every mass extinction, every climate change, every ecological upheaval. It did so by being undemanding, flexible, metabolically cheap. That's success by any reasonable measure. Not the kind of success that gets celebrated. No one writes popular books about Trichoplax, but real lasting success measured in survival. Maybe some Ediacaran descendants are equally successful. We just don't know it yet. Let me take you to the Nama group in Namibia one more time, because what those rocks show is crucial for understanding how the Ediacaran ended. These are sediments spanning the terminal Ediacaran and earliest Cambrian, giving us a continuous record of the transition. And what they show is not sudden catastrophe, but gradual replacement. In the oldest layers, classic Ediacaran forms dominate. Rangeomorphs, Dickinsonia-like organisms, Pteridinium, the full assemblage. The communities look distinctly Ediacaran, soft-bodied, mat-associated with no evidence of predation. Move up through the sequence and you start seeing changes. Small shelly fossils appear, organisms with mineralized parts, but not yet full shells. Early arthropods show up with jointed appendages and segmented bodies. Trace fossils become more complex, showing deeper and more elaborate burrowing. For a while, the two worlds coexist. Ediacaran forms and Cambrian forms are found together in the same rocks, representing the same time intervals. But gradually, the proportions shift. Ediacaran organisms become rarer. Cambrian organisms become more abundant and diverse. By the time you reach unambiguous Cambrian rocks, the Ediacaran organisms are gone. Completely. The assemblage is entirely Cambrian. Arthropods, mollusks, brachiopods, early echinoderms. Hard-shelled, mobile with complex organ systems. The world has transformed. This pattern, gradual replacement rather than sudden extinction, suggests competitive displacement. The Ediacaran organisms weren't killed by a catastrophe. They were out-competed, slowly, over millions of years, by organisms that had evolved features better suited to the changing conditions. What features? Shells, providing protection from predators. Guts, allowing efficient processing of food. Nervous systems, enabling fast responses and complex behavior. Active mobility, allowing exploitation of patchy food resources. All the innovations that define modern animals. Ediacaran organisms had none of these. They were adapted to a world without predation, with abundant dissolved nutrients, with stable mat-covered substrates. When that world changed, they couldn't keep up. Not because they were poorly designed, but because evolution had moved on. This is how extinction usually works. Not dramatically, but incrementally. One lineage reproduces slightly faster, uses resources slightly more efficiently, tolerates environmental fluctuations slightly better. Over generations, that slight advantage compounds. The less competitive lineage shrinks, becomes rarer, is pushed into smaller and more marginal habitats, and eventually disappears. It's not the stuff of disaster movies. It's quiet, slow, inexorable, but it's how most species go extinct. And it's what happened to the Ediacaran organisms. They were outcompeted gradually and completely by organisms whose innovations were better matched to Cambrian conditions. There's a melancholy to this. The Ediacaran organisms were successful for nearly 100 million years. They created ecosystems unlike anything before or since. They filled ancient oceans with strange and beautiful forms. And then, the world changed, and they couldn't adapt fast enough. Not because they failed in any meaningful sense, but because evolution never stops, and what works today might not work tomorrow. Now let's talk about something that will make you reconsider your place in this story. You are not an Ediacaran organism. Your lineage split from the Ediacaran forms, assuming any connection at all, more than 500 million years ago. You're descended from the organisms that outcompeted them, from the Cambrian victors, not the Ediacaran losers. Your ancestors were probably small, probably worm-like, probably living in sediments or on the seafloor during the Ediacaran. They survived the transition. They evolved shells, or burrowing behavior, or fast locomotion, whatever was necessary to persist as the world changed. They joined the arms race. They adapted to predation. They became the organisms that would eventually diversify into vertebrates. Somewhere in your deep ancestry, there's a lineage that navigated the Ediacaran Cambrian transition successfully. Not the Dickinsonia lineage. That ended. Not the ranger morphs. They're gone. But something. Some bilaterally symmetrical, probably unseen organism that we don't have fossils of, that survived and reproduced and gave rise to descendants that eventually included you. You are connected to the Ediacaran world. Not to the famous forms we've been discussing, but to the ecological milieu they inhabited. Your ancestors lived in those oceans, breathed that low-oxygen water, crawled on those microbial mats. They experienced the rise of predation. They felt the change as the world transformed from Ediacaran to Cambrian. And they survived. Most lineages didn't. Most Ediacaran experiments ended. But yours didn't. Your lineage persisted through every subsequent crisis, every mass extinction, every climate change, every ecological upheaval for the next 540 million years, all the way to this moment. That's the power of evolutionary adaptation. Not perfection, not inevitability, just persistence. Your ancestors weren't the strongest or the smartest. They were the ones that survived long enough to reproduce, generation after generation, through conditions we can barely imagine. Think about what that means. 540 million years. That's 540 million trips around the sun. Billions of generations. Countless opportunities for the line to end, and it didn't. Through the Cambrian Explosion, through the Ordovician radiation, through the end Permian extinction that killed 90% of marine species, through the asteroid that ended the dinosaurs, through ice ages and hothouse climates and supercontinent assemblies and breakups. Your lineage made it through all of that. And now you're here, thinking about the Ediacaran organisms that didn't make it, wondering what lessons their disappearance holds. Here's one lesson. Success is temporary. Dickinsonia was successful for tens of millions of years. Then it wasn't. Humans have been successful for a few 100,000 years. Will we still be here in tens of millions of years? Probably not, at least not in a form we'd recognize. Species don't last forever. Lineages continue, but individual species are transient. This should inspire both humility and wonder. Humility because we're not special in any ultimate sense. We're one experiment among countless others, and most experiments eventually end. Wonder because despite the odds, despite the extinctions and the upheavals, and the sheer randomness of existence, life persists. Not any particular form of life, but the process itself, the endless trying, the adaptation, the continuation. The Ediacaran organisms are gone. Their strange body plans have been extinct for over 500 million years. But their story continues, in the rocks that preserve their impressions, in the scientific questions they raise, in the lessons they teach about adaptation and extinction. And in a deeper sense, their story continues in you. You exist on the same planet, following the same laws of chemistry and physics. You're part of the same unbroken chain of life that includes them, even if your branch split from theirs 500 million years ago. The carbon atoms in your body have been recycled through countless organisms. Some of those atoms might have been part of Ediacaran organisms once. Matter cycles, energy flows, life continues. The Ediacaran ended, but it made the Cambrian possible. The Cambrian made vertebrates possible. Vertebrates made mammals possible. Mammals made primates possible. Primates made you possible. The chain is unbroken and every link was necessary. Remove the Ediacaran and you don't exist. Not because you descended from Dickinsonia, you probably didn't, but because the Ediacaran was part of the sequence that led to you. History doesn't have alternate paths we can explore. We can't know what would have happened if the Ediacaran organisms had survived, if the Cambrian arms race had never started, if predation had never evolved. Maybe intelligence would never have arisen. Maybe complex brains require the selection pressure that comes from predator-prey dynamics. Maybe a world of peaceful osmotrophs would never produce organisms capable of thinking about their own history. Or maybe it would have eventually through different paths. We can't know. We only have the one history, the one that actually happened. And in that history, the Ediacaran organisms disappeared, the Cambrian organisms thrived, and eventually you appeared, capable of looking back through deep time and wondering about the roads not taken. That capacity to think about other possibilities, to imagine worlds that don't exist, to ask questions about extinct organisms you'll never see, is itself a product of evolution. It emerged because it was useful. Thinking about the future, modeling what might happen, requires the ability to imagine scenarios that aren't present. And that ability, once it exists, can be turned backward as well as forward. You can imagine the past. You can reconstruct vanished worlds. You can think about Ediacaran organisms and what their existence means. This is a remarkable thing. Matter organized itself into forms capable of understanding their own history. Carbon and hydrogen and oxygen and nitrogen, following physical laws, producing organisms complex enough to contemplate deep time. The universe looking back at itself through your eyes, thinking about forms it tried 500 million years ago and then discarded. The Ediacaran organisms never thought about themselves. They didn't wonder about their origins or worry about their eventual extinction. They just lived, generation after generation, for nearly 100 million years. And then they were gone. You do think about yourself. You can wonder about your origins. You can worry about eventual extinction, both personal and species-wide. Does that make you better than them? More successful? Or just different? Maybe success shouldn't be measured by duration alone. Maybe it should include experience, awareness, the capacity to reflect on existence. By that measure, you succeed in ways Ediacaran organisms never could. But they succeeded in ways you never will. They existed without fear, without anxiety, without the burden of self-awareness that makes you capable of asking whether your existence means anything. Both strategies work in their own contexts. Both are products of evolution exploring possibility space. Neither is inherently superior. They're just different solutions to the problem of being alive. The Ediacaran solution lasted 100 million years. Yours has lasted a few 100,000. Time will tell which approach has more staying power. But perhaps staying power isn't the right metric. Perhaps what matters is the experience of being alive, the complexity of awareness, the richness of existence in the time you have. The Ediacaran organisms had nearly 100 million years. They never knew they had it. You have decades if you're lucky. But you know, you're aware. You can think about your place in deep time, your connection to vanished worlds, your role in a story that spans billions of years. That awareness is precious. Use it well. Thank you for taking this journey through deep time with me. If this exploration has meant something to you, please subscribe. Share this with someone who might find it meaningful. Leave a comment about what surprised you, what moved you, what made you think differently about life and time, and your place in the vast sweep of history. The Ediacaran happened. It was real. Strange organisms lived in ancient oceans, thrived for longer than you can truly comprehend, and then vanished, leaving only impressions in stone and questions in our minds. You're connected to that story, not through those specific organisms, but through the world they helped create, the changes they experienced, the transition they didn't survive. The story that began in the Ediacaran continues in you. You're a chapter in something vast and ancient and ongoing. The organisms that come after you in 10,000 years, in 1,000,000 years, in 10 million years, will be different from you in ways you can't imagine, just as you're different from Ediacaran organisms in ways they couldn't have imagined. But the process continues, life adapting, changing, exploring new forms, trying new strategies. Some will work for a while, most will eventually fail. A few will persist long enough to give rise to lineages that continue the story. You're part of that story right now. This moment, you, here, thinking about Ediacaran organisms and what they mean, is part of the ongoing narrative of life on Earth. It matters, not because it's permanent, but because it's happening. You're alive, aware, capable of wonder. That's enough. Sleep well tonight, knowing you're connected to 500 million years of history. The Ediacaran organisms couldn't know their place in the story, but you can. You can understand that you're a temporary expression of processes that are far older than you and will continue long after you're gone. You can find meaning in that connection, or peace in that perspective, or simply wonder at the strangeness of being a thinking thing in a universe that mostly isn't. The Ediacaran taught us that success is temporary and contingent. That adaptation to one world doesn't prepare you for the next. That most experiments eventually end, and that's not failure. It's just the nature of evolution. You're an experiment too. Make it a good one while it lasts.